So this is the first discrete question of the biochemistry section of full length 5. So this is question 11, and it states which amino acid exhibits a beta branched side chain, a beta branched side chain. So what does that mean? A beta branched side chain. So let's take a look at the average structure of an amino acid, right? So here we go, and then let's say we have some kind of R group here. Let's actually extend this R group out, and then let's do something like this. Now, what we have here is the alpha carboxyl group, the carboxylic acid group, and then here we have the amine group. So that's the alpha amine group. And so what that means is this carbon right here, this is actually the alpha carbon, and that makes this carbon right here the beta carbon. So when it says beta branched, it means it has to be branched at the beta section, or at the beta carbon. So the first answer choice is alanine, and because I'm lazy, I've already just copied and pasted all the amino acids here. So this is alanine, and you guys should know this by now. Remember the amino acid structures, the names, the abbreviations, all of that, super important on the exam. So this is alanine. We see the amine group, the carboxylic acid group. Like I said, this is the alpha, and then this means this is the beta. And you'll notice there's no branching here, so it can't be alanine. If we move on, this is going to be leucine. There we go. So now leucine, again, same idea here, alpha, and this is beta. And you might think this is branched because now you have another carbon attached to this beta carbon, but branched means there has to be one more carbon here, okay? There has to be another carbon attached to this beta carbon. Here you just see it's kind of linear, just going from one carbon to another. For it to be branched, there has to be some kind of other carbon attached to this beta carbon as well. So it can't be this leucine either. Now C says isoleucine. So same thing here, this is the alpha, this is the beta. Now you'll notice here on the beta carbon, you have one carbon here and it's branched out. There's another carbon that's attached here. Now because there's two groups here, that means this beta carbon is branched, making the isoleucine. This seems like a pretty good answer for a beta branched amino acid, right? The beta carbon is branched. So this looks like it checks out so far. Let's take a look at the last answer choice, and it says glycine. So we know this is glycine. Now, you guys should know by now that, here's the carboxylic acid. I mean, this is the alpha carbon. There's not even an R group attached. Well, I guess the R group is the hydrogen, but you'll notice there's not even a beta carbon to begin with here, meaning glycine can't even be the answer here. So that makes C the correct answer for this problem. So this is question 12, and it says, in contrast to chemical messengers in classical endocrine signaling systems, chemical messengers in paracrine and autocrine signaling systems are blank. So what does this mean? So we see three different signaling systems here. We've got to first figure out what those are. And so you guys should know this by now, but endocrine, what that essentially means is you have some kind of source of this signaling molecule in your body, and it releases these factors, these endocrine signaling factors. Let's label them orange, like this. And it actually travels through the bloodstream. It gets carried away in your blood, okay? So it enters your bloodstream, it gets carried away, and it actually affects tissues distance from the source. It can affect tissues distantly from the source. So that's what endocrine means. Paracrine is a little bit different in that, let's say this is my source cell. Let's say it's releasing these paracrine factors. It's going to actually only act very locally to its receiving cell. So let's say this is a receiving cell here, this is the source cell here, and this paracrine, the signaling molecule, it's only going to act on the nearby cells in a very local environment. And so finally that leaves us with autocrine. What does autocrine mean? Autocrine means if this is the source, it releases the molecule, and it's actually going to come back and affect the same source. So the source, source cell acts as both the source and the receiver of the signal. That just means the signal acts on itself. So that's what these mean. Now let's take a look. A says these paracrine and autocrine signaling systems are secreted by neurons only. That's not true. Paracrine and autocrine signaling systems are found with all other cell types in the body, not just the neurons. So we know that A is just factually incorrect. Paracrine and autocrine signaling systems are not likely to bind cell receptors. Paracrine, autocrine, endocrine signaling systems all release factors that can only act by binding onto cell receptors, whether that cell receptor be inside of the cell, the receiving cell, or on the surface of the receiving cell. So B is incorrect. Paracrine and autocrine signaling systems are usually transported by ducts. That's not true. Ducts transport exocrine factors. So exocrine factors are things like sweat, saliva, um, milk, things like that. That's what the exocrine factors are. And paracrine, autocrine, and endocrine signaling systems, none of them are transported through this exocrine pathway. So finally, that leaves us with not transported by blood vessels. So like I told you guys, endocrine signaling systems, they have to act far from where the source is. So that means those molecules, the signaling molecules, are going to be carried away in your blood to reach other tissues in your body. On the other hand, paracrine and autocrine signaling systems, they don't need to be carried by blood because the receiving molecule or the receiving cell is right next to the source cell, or the receiving cell is the source cell itself. So there's no reason for these molecules to be carried away by the blood vessels, which means D is our correct answer. So this question says, which membrane transporter is electrogenic, and translocates a net charge across the membrane? So it's saying, okay, let's say we have our membrane here, and how do we get a net charge, positive, or maybe it's negative, across the membrane, okay? So the first one says NaH exchanger. Now, what does that mean? Let's say I have this membrane here, Na plus on the outside, and H plus on the inside. Exchanger means this protein is going to exchange the Na plus and the H plus. So this guy's going to come here, and this guy's going to go there. Well, what you notice is that you're just swapping these positive charges, so there's not a net charge that's being translocated. So that makes A incorrect. Now, B says Na plus Cl minus co-transporter. So what's a co-transporter? That's when you have a protein that moves these molecules in the same direction. So let's say I have Na plus here, chlorine here. Let's say this is my protein. It's going to bring in both the Na plus and the chlorine in. So what does that mean? So one Na plus and one Cl minus, net charge is 0. And when it brings it into the inside of, or maybe it's the outside, when it brings it across the membrane, the net charge is still going to be 0 since you have, again, one positive charge and one negative charge. So it can't be B. Now C says Na plus and a glucose co-transporter. So then, let's see, we have our membrane, Na plus, and let's say we have our big molecule of glucose here. Well, if you bring in Na plus and also bring in glucose, well, you've just translocated, right, one net charge, one positive charge. So the glucose, its overall charge is 0, but the Na plus, it has a positive 1 charge, right? So on the outside, it used to be +1, but now that you co-transported it, now the inside, you have a net charge of +1. So that makes C the correct answer. And GLUT2 facilitative glucose transporter, let's take a look at D. What does that mean? Well, this one is just something that brings in glucose or takes out glucose. So let's say I have my glucose molecule here, it's just going to come inside like that. And you'll notice, again, glucose has a net charge of 0, so no matter how glucose moves out or in, there is no translocation of a net charge across the membrane, meaning D cannot be the correct answer. Again, leaving us with C as the correct answer. Okay, so this is another discrete question. It says, which type of cell has a plasma membrane, lack of a nucleus, lack most organelles, and are shaped like a flat disc with a concave center? All right, so this part right here, the flat disc, concave center, that's a pretty important key right there, and the lack of nucleus as well. So if you'll notice these, this is half, discrete, as in you need to know characteristics of different cell types, and the other half is kind of like a vocab quiz, So erythrocytes, these are actually red blood cells, okay? And so I'll just move down the line here, and let's see. So erythrocytes, do they lack a nucleus? Or I guess first, do they have a plasma membrane? Yes. Do they lack a nucleus? Yes. Do they lack most organelles? Yes. And are they shaped like a flat disc with a concave center? So most of you guys probably know by now what these red blood cells look like, right? They're like flat discs, and they have like this smushed in center. And so that these all fit the description of erythrocytes perfectly. So this is a pretty good answer so far, but let's look at the rest. Fibroblasts, do they have a plasma membrane? Yes. Do they lack a nucleus? No. Fibroblasts do have a nucleus, which means it can't be B. Monocytes, do they have a plasma membrane? Yes. Do they lack a nucleus? No. Can't be C. Neurons, do they have a plasma membrane? Yes. Do they lack a nucleus? No. Matter of fact, they have a nucleus, they have most organelles, and they are not shaped like a flat disc with a concave center, which means D cannot be correct, leaving A as the correct answer for this discrete.